A sterile room. Hums under ultraviolet lights. A tooth is ground to powder. Gloved hands steady over a centuries-old skull. The bitter tang of bleach thick in the air. For decades, we believed the Americas had a single origin story. One migration, one timeline, one people. There was no hidden branch, no vanishing lineage, no replacement of entire populations. But there is just one problem. Ancient DNA refuses to fit that tidy map. The hard evidence, Anzick one showing Siberian ancestry, the Upward Sun River infant revealing a distinct ancient Beringian lineage, and a vanished genome from ancient Bogota in Colombia, demands we rewrite everything we thought we knew about the origins of the first Americans. So does the real story begin with one founding people, or are there multiple origin threads from the start? Gloved hands lift a brittle bone. A fine white powder drifts into the air. Stainless steel tweezers click against glass. A UV lamp throws tiny scratches into sharp light. The clean room smells of ethanol and cold metal. That powder is a time capsule, and it asks a single stark question. Did the peoples of the Americas descend from one founding migration, or did multiple distinct threads arrive and shape the continent from the start? Everyone knew the tidy map. Textbooks traced a single bold arrow from Siberia into the heart of the Americas. The story was simple. One migration, one founding group crossing Beringia as the glaciers retreated, their descendants fanning out to fill an empty continent. The Clovis culture, with its distinctive spear points, stood as the first widespread signature, proof of a clean beginning around 13,000 years ago. There is just one problem. There was no hidden branch, no vanished community, no sweeping replacement, no flawless single highway into the past. What was missing was a witness that could not be moved or misread, a record written in the bodies of the ancient dead themselves. Ancient DNA arrived and changed how we read the map. The Anzick one Clovis-era genome shows the Clovis people were closely related to indigenous peoples of the Americas, consistent with Siberian ancestry and not evidence for a European Solutrean origin. That finding is a clear answer to one controversial claim. The Upward Sun River infant genome revealed a separate and deeply divergent line now called ancient Beringian. That infant suggests deep population structure in Beringia and an early split that led to later Native American branches, rather than a single undifferentiated wave. Across the Americas, ancient genomes support an early divergence and a surprisingly rapid spread, followed by regional changes through time. New results from the Bogota Altiplano in Colombia found a previously undocumented lineage that later disappears from the genetic record, replaced by other groups. Those data show the peopling story includes turnovers and regional replacements, not just one uninterrupted line. Archaeology keeps pressing the timeline too. Sites like Monteverde in Chile and the very early White Sands footprints force genetic models to account for presence in the Americas as early as the late Pleistocene. Genetics and archaeology must be read together. We must be clear. Genetics is not the same as identity. DNA does not decide who indigenous people are, and genetic results do not replace oral histories, cultural continuity, or the rights of living communities. Ancient DNA is one powerful line of evidence among many. Some conclusions are widely supported. The Anzick one genome ties Clovis to indigenous ancestry with Siberian roots. The ancient Beringian signal at Upward Sun River shows deep early structure. Ancient genomes point to an early divergence and rapid dispersal, and later regional shifts are real. Other questions remain debated, including the precise routes people took across ice and sea, the exact timing and number of migrations, and the extent of later gene flow across regions. The old textbook map is no longer enough. The past looks messier and more interesting. Either ancient DNA confirms one founding story that later splintered and diversified, or it shows multiple origin threads from the start. In a white, clean room, a gloved hand brushes bone dust into a tray. The hiss of a decontamination spray fogs the air. A single hair clings to a vial. A bead of condensation crawls down a cold tube, and fluorescent lights hum over stainless steel. This small scene reframes the whole story. Because does the DNA point to one tidy founding, or does it reveal many threads braided together from the start? Everyone knows the story. A single group crossed from Siberia into the Americas, spread south, and became the first peoples of the New World. That simple migration map was taught with confidence, a single line on a map that seemed to explain everything. There is just one problem. DNA has become a new kind of witness. DNA is a fragile witness. Most of it vanishes in a few thousand years, shredded by heat, water, and microbes. Only in rare places, cold caves, frozen tundra, and dry rock shelters does a trace survive. Even then, a stray skin cell or a single breath at the wrong moment can drown the signal. That is why ancient DNA labs look like operating rooms. Researchers suit up in full body gowns, pass through airlocks, and work under filtered hoods. Bone powder is sifted, chemicals tease out molecules, and machines scan for the faintest hints of genetic code. Most samples fail. When a genome does reveal itself, it is a time-stamped receipt, proof of who was present, how they were related, and when their branch split from others. DNA cannot tell us what language people spoke or what stories they believed. DNA is not identity, but by comparing ancient genomes, scientists can build a family tree with real branches and forks. There was no single, unbroken march. It was not only one route. It did not come from Europe as the old Solutrean idea proposed, and it was not immune to later turnovers. The evidence that rewrites the tidy map comes in genomes. The Anzick 1 genome, from the Clovis era, shows that Clovis people are closely related to indigenous peoples across the Americas, and that their ancestry is consistent with Siberian roots, not a European origin. That finding collapsed an old hypothesis, and it anchored Clovis inside the broader Native American family. At Upward Sun River in Alaska, an infant genome revealed a distinct ancient Beringian lineage. That genome suggests deep structure in Beringia, an early split that left one branch isolated in the north and other branches that later became the ancestors of most Native Americans. The word ancient Beringian marks a major fork in the tree. Across the continents, more ancient genomes tell a pattern of early divergence and rapid spread, followed by regional changes and later gene flow. In some places, ancient lineages persist. In others, they vanish or are replaced. A recent study from the Bogotá Altiplano in Colombia found an unexpected, previously undocumented lineage that later disappears from the archaeological record. A clear sign that peopling included turnovers, not just a single uninterrupted line. The finding from Colombia shows that replacement and regional shifts are part of the story. Archaeology presses on the same questions. Very early footprints at White Sands in North America suggest people were on the landscape earlier than many models expected. And that timeline challenges genetic estimates and forces new hypotheses about movement and population structure. What is widely supported is this: the first peoples of the Americas trace large part of their ancestry to Siberian sources. Early splits occurred in Beringia or nearby, and a rapid spread seeded much of the continents. What is still debated includes the precise routes people took, the exact timing of splits, the number of distinct migrations, and the extent of later gene flow between regions. Genetics is not a definition of identity. Living indigenous communities define themselves through culture, language, and lived relationships. Ancient genomes are scientific data that illuminate past population events. They do not determine present-day belonging. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. Gloved hands lift a small box under sterile lights. Powdered bone drifts like fine ash. A thin smear of ochre stains the cloth. Cold metal tongs click against brittle bone. The lab smells faintly of bleach and old earth. One discovery will reframe everything. Did the makers of Clovis tools come from across the ocean or from deep across Beringia and Siberia? Everyone knew the neat story. People crossed a single bridge, moved in a single wave, and spread a single culture. That story sounded settled. There is just one problem. There was no sudden European arrival. There was no single simple path. It wasn't a straight line. DNA became a new witness, and it changed the testimony. The Clovis child, known as Anzick I, was found in 1968, buried with more than 100 ochre-stained stone tools. In 2014, Eske Villerslev's team sequenced the child's genome, and the results were clear. The child lived around 12,600 years ago, and his DNA clusters tightly with indigenous peoples of the Americas, not with Europeans or with East Asians. The genome also carries a distinctive signal from an ancient Siberian group called Malta, showing that part of the ancestral Native American line traces back to Siberia long before people set foot in the Americas. That finding closes the door on the Solutrean hypothesis, the idea that Ice Age Europeans peopled North America by following the ice edge. Anzick I stands as direct evidence that the people who made Clovis tools were rooted in Beringia and Siberia, and that Clovis traditions belong to the indigenous story of the Americas. But the picture is more complex than a single genome can capture. The Upward Sun River. Infant genome revealed a distinct ancient Beringian lineage, which suggests deep population structure in Beringia and an early split that led to later Native American branches. Ancient genomes from across the continents show an early divergence and rapid spread, and later regional shifts. A recent ancient DNA study from the Bogotá Altiplano in Colombia uncovered a previously undocumented lineage that later disappears and is replaced, proving that population turnovers happened, not just unbroken continuity. What changed is our model. We moved from a tidy migration map to a layered story of early splits, rapid dispersals, and later regional turnover. What did not change is this fact: DNA does not decide identity. Genetic results do not define who indigenous peoples are. Those identities are lived, cultural, and political. What remains debated includes precise routes, exact timing, the number of distinct migrations into the Americas, and the scale and timing of later gene flow. New archaeological finds, like very early footprints, press genetic models to account for earlier presence on the continents, and that dialogue between bones and genomes is ongoing. At stake is a simple choice about the origins of the first Americans. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. A cold room under bright lamps, a stainless steel table rimmed with frost, fine bone powder settled like talc, ochre dust staining gloved fingertips, a tiny rib, almost translucent, laid out with a probe. One discovery will reframe everything, so was this a single crossing or many threads woven together? Everyone used to tell the same story, confident and neat. A single migration moved across a land bridge, people spread south, and the map was simple and final. There is just one problem. The DNA speaks with a different voice. There was no single route. There was no single timing. It wasn't a single uninterrupted line. In 2010, archaeologist Ben Potter and his team excavated Upward Sun River in Alaska, where 2 infants were buried with ochre and hunting tools, nearly 11,500 years ago. Genetic sequencing of one infant revealed a surprise. This child belonged to a distinct ancient Beringian branch, a lineage that split from the ancestors of other Native American groups around 20,000 years ago, and that lingered in Beringia for millennia. That genome shows Beringia was not just a bridge for passage. It was a place where people lived, diverged, and sometimes vanished from the genetic record. The Upward Sun River genome makes the peopling of the Americas look less like a single thread and more like a branching tree that begins high in the north. At the same time, the Clovis era. Anzick. One genome demonstrates something else important. The Clovis child is closely related to indigenous peoples across the Americas, and the genetic signal is consistent with Siberian ancestry, not a European Solutrean origin. That result is now widely supported by peer-reviewed research, and it helps close one long-standing debate. Across the continent, ancient genomes point to an early divergence and a rapid spread southward, followed by later regional shifts. A recent ancient DNA study from the highlands near Bogotá in Colombia found a previously undocumented lineage that appears and then disappears, replaced by later groups. These findings show that population turnovers occurred, not just a simple uninterrupted descent. What is widely supported is clear. Ancient genomes confirm deep structure in the north, an ancient Beringian split, and strong connections between Clovis-era people and later indigenous populations. What is still debated includes the exact routes, the precise timing, how many distinct migrations there were, and the extent of later gene flow between regions. Very early archaeological finds, like the White Sands footprints, continue to press timelines that genetics must account for. Genetics is powerful, but it does not define identity. DNA is a scientific record of ancestry and movement, and it must be read alongside archaeology, oral histories, and living communities. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. Gloved hands in a clean room, bone powder like talc drifting on a tray, frosted tubes humming in a freezer, tiny teeth fragments catching a harsh fluorescent glare, a single hair-thin crumb of ancient collagen brushed into a vial. What if ancient DNA shows not one clean migration, but either a single founding wave that later splintered or multiple origin threads from the start? Everyone knows the story. A simple map of a single migration across Beringia, a spread of Clovis people, a neat ancestry that seeds the whole continent. There is just one problem. DNA has become a new witness, and it refuses a tidy map. There was no European Solutrean origin, there was no single route, there was no uninterrupted line from first arrivals to every later population. The Anzick, one genome from the Clovis era, is a clear piece of evidence. It shows Clovis people are closely related to indigenous peoples of the Americas, and it is consistent with Siberian ancestry, not a European origin. This finding is widely supported, and it undercuts one long-standing hypothesis. The Upward Sun River. Infant genome introduced another surprise, a distinct ancient Beringian lineage that split off deep in Beringia. And that split is part of the reason Native American ancestry is structured from the very beginning. Ancient genomes across the Americas now point to an early divergence and a rapid geographic spread, followed by later regional change and mixing. The recent ancient DNA study from the Bogotá Altiplano in Colombia adds a dramatic southern counterpoint. Researchers recovered a lineage that branched early, persisted in the highlands for thousands of years, and then disappears from the record as new populations sweep through. Genomes from other sites tell the same story, some lineages holding ground for millennia, others fading or being absorbed. This is not a script about who people are. Genetics is not identity. Genetic data reveal patterns of ancestry and movement, not the totality of culture and belonging. Some findings are widely supported, such as the Anzick one result, the ancient Beringian split, and the broad pattern of rapid early divergence. Still debated are the exact routes people took, precise timing, the number of migrations, and the extent of later gene flow that reshaped regional populations. Reconciling very early archaeological evidence like the White Sands footprints with genetic models is an active part of that debate. What changes is the story we tell. What stays is the fact of deep time and complex human choices. The peopling of the Americas looks less like a single highway and more like a braided river of movement, pause, and replacement. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. In a clean room, under a pale fluorescent light, gloved hands tilt a tiny vial, bone powder mists a metal tray, small glass tubes click on stainless steel, and the low hum of freezers fills the air. Ancient DNA makes the bone itself a witness that can reroute the origin story of the Americas. Did the first Americans arrive in one founding wave, or did many origin threads arrive from the start? Everyone knows the story. A single migration, a clean coastal route, Clovis at the beginning, and everything else following in a straight line. There is just one problem. Ancient genomes are the new witness, and they tell a different, messier truth. There was no European Solutrean origin. It was not a single unbroken march south. There was no simple, uninterrupted line. Still, some clear conclusions are widely supported. The Anzick I Clovis-era genome shows Clovis people are closely related to indigenous peoples across the Americas, and it is consistent with Siberian ancestry rather than Ice Age Europeans. The upward Sun River infant represents a distinct ancient Beringian lineage, evidence of deep population structure in Beringia, and an early split that preceded the branches that formed later Native American lineages. Ancient genomes sampled across North and South America support an early divergence and a rapid initial spread, followed by regional changes over time. New results complicate that picture further. A recent ancient DNA study from the Bogotá Altiplano in Colombia uncovered a previously undocumented lineage that later disappears or is replaced, showing that turnover and local replacement are part of the story. Very early archaeological evidence, including the white sands footprints dated to around 21,000 years ago, pressures the timelines that genetic models must explain and sparks lively debate about how fast populations moved and when. What remains debated are the details. Scientists disagree about the number of migrations, the relative importance of coastal versus interior routes, the precise timing of splits, and the extent of later gene flow between groups. These are active research questions, not contradictions of the basic finding that ancestry links the Americas to Siberia. One boundary must be respected. Genetics can trace population history, and genetics can reveal splits, mixtures, and replacements. Genetics does not define culture, language, or who belongs. Identity is claimed and lived, not read from a genome. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. Gloved hands lift a brittle rib, bone dust drifting like ash in the sterile light, a tray of tiny teeth clicking against cold metal, a faint chemical smell of ethanol, and the steady, distant hum of a freezer. What that quiet scene forces us to ask is this. Either the first peoples of the Americas began as a single founding population that later split into many branches, or they arrived as multiple threads, weaving, mixing, and sometimes replacing one another. Which is it? Everyone knows the tidy story. One migration, a single walk across Beringia, a clean march into the continents, and then a steady spread that became the indigenous peoples we know today. That version is confident, easy to draw on a map, and it comforts a sense of order. There is just one problem. There was no single road. There was no simple march, there was no European origin, and there was no uninterrupted line. Ancient DNA has become a new witness, and it speaks in genomes, not maps. The evidence rewrites key parts of the tale. The Anzick 1 genome from a Clovis-era burial shows Clovis people are closely related to indigenous peoples of the Americas, and it is consistent with Siberian ancestry, not a European Solutrean origin. The upward Sun River infant represents a distinct ancient Beringian lineage, which suggests deep structure in Beringia and an early split that led to later branches. Ancient genomes recovered across the continents point to an early divergence and a rapid spread, followed by regional shifts and later turnovers. And a recent ancient DNA study from the Bogotá Altiplano in Colombia found a previously undocumented lineage that later disappears or is replaced, showing that the peopling story includes population turnovers, not just one uninterrupted line. For context, archaeological finds such as the very early footprints at white sands press on the timelines that genetic models must explain. But genetics and archaeology answer different questions. Genetics is not identity. DNA does not decide who indigenous peoples are, and genetic relationships do not replace cultural continuity, oral histories, or modern identities. Some conclusions are now widely supported. Anzick 1 links Clovis to indigenous American ancestries and to Siberian roots. The upward Sun River infant demonstrates ancient Beringian ancestry and early structure. Ancient genomes show early splits and a rapid spread, plus later regional change. What remains debated is the exact timing, the precise routes, the number of migrations, and the extent of later gene flow and replacement. The choices we make about which threads to follow shape the story we tell. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start. Ancient DNA proves the first American story is not a simple lineage. It is layers of arrival, loss, and change. Today, every new sequence challenges what we think we know. The past is not settled. It is still being rewritten, strand by strand. There was no single route, no single timing, no simple map. Either ancient DNA confirms one founding story that later splintered, or it shows multiple origin threads from the start.